Hello, everyone. I want to start off by thanking our sponsor, the Tim Gard Distinguished Lecture Series, brought to you by the Friends of the Waikiki Aquarium. We at St. Andrews Schools, the Prior, are happy to be the hosting sponsor, and thanks to media sponsor Hawaii Public Radio. Just for some housekeeping, this is a pre-recorded lecture with some live questions and answers starting at 5:15. We will be recording tonight, and it will be posted on the Friends of Waikiki Aquarium website in December. Everyone will be muted throughout the presentation. We welcome any questions during the presentation via chat. We will also collect questions throughout and ask the speakers during the live Q&A. Please note which speaker you're posing the question to. Please be sure that your video or camera is disabled during the presentation. If enabled, everyone attending will be able to see you and you may appear on the recording. Now, I would like to welcome our two speakers, Dr. Emmett Duffy, Director of the Smithsonian Institution's Tennenbaum Marine Observatories Network, which is also joining us from Virginia, and Dr. Mary Hagedorn, who is a senior research scientist with the Smithsonian Conservation Biology Institute. Dr. Hagedorn is also working here in Hawaii Institute of Marine Biology on Coconut Island. As someone who is interested in marine science, I am intrigued by how these scientists became experts in this field and what their research in Kāneʻohe Bay can tell us.
Hello, everyone, and thanks for joining us. My name is Emmett Duffy. I'm a marine biologist at the Smithsonian Institution in Washington, D.C., and it's a great honor to deliver this distinguished lecture to the Friends of the Waikiki Aquarium, along with my friend and colleague, Dr. Mary Hagedorn of the Smithsonian Conservation Biology Institute, who will be talking directly after me. So I want to talk to you today about the meaning of life. Not that meaning of life with the guru on the mountaintop, although I think our topic today is in fact important to that broader meaning of life. What I mean is our connection to the rest of the living world, how it's essential to our own human well-being, and how those connections are changing in the modern world and what we can do about it. I especially want to consider these issues in the context of the unique marine ecosystem of the Hawaiian Islands. So we human beings are living creatures like the fishes and birds and insects and plants that surround us. Obviously, we are unique and special in many ways and different than the other forms of life, but we're also similar in that people depend on other living things for our own survival and health and happiness. People are part of nature and connected to the rest of the ecosystem in many ways. In scientific language, we say that we depend on biodiversity. That is this rich array of living organisms around us. That is for food and economic security, the fishes that we eat, for our health through medicines that are derived from natural products, for livelihoods, and also to nourish and nurture the spirit. Over the last 20 years or so, a large body of scientific evidence has been collected showing that this diversity, this rich variety of life, is actually important to the continued health and stability of the ecosystems that we depend on in many ways. Unfortunately, that rich web of life is in trouble. A major synthesis by scientists all around the world recently concluded that 1 million species of plants and animals are in danger of extinction during the lifetimes of children that are young today. So what do we do about that? And thinking about tropical ecosystems in particular, there has been a lot of attention to major changes in the ocean recently. A warming ocean has caused serious injury, for example, to coral reefs as a result of the decades of fossil fuel burning that we've done. For example, over the last 5 years, the Great Barrier Reef of Australia has bleached, meaning that its corals have died, killing large areas of the reef. How do we fix this precious heritage and save it? Well, we can't fix it if we don't know how it works and where it is. And so the first challenge that we have in saving the ocean is that for much of the tremendous biodiversity on Earth, we know little or nothing about where it is or how it's working. So this was the motivation for the Smithsonian's Marine Global Earth Observatory, or MarineGEO program, that was launched about 7 years ago now. MarineGEO is a global community of partners and a platform for doing the kinds of research that we need to inform conservation and better management. So we seek to understand how coastal marine ecosystems work so that we can help keep them working. And we do that by using a common toolkit that is the same kinds of scientific methods and protocols wherever we go, so that we can produce a global picture of the health of marine life. And basically, MarineGEO recognizes that we can do much more together than we can alone. It is a community. The network is people working together. So our premise is that a global picture of the ocean's biodiversity will succeed by organizing people around common interests. 1,000 heads are better than one when they're working together. We can think of this as a way of gathering the knowledge we need to provide healthcare for the ocean. We can't understand how to help a sick patient if we don't know their vital signs, how their bodies are working, and therefore what condition they're in. And we need diagnostic tests to understand what is causing changes in condition. So what MarineGEO aspires to do is we build infrastructure and capacity that is training people, bringing people together to learn the same techniques so that we can learn more together. We support long-term monitoring of vital signs. We like to say that we take the pulse of coastal ocean life, and we conduct experiments to diagnose what are the causes of the changes that we see. And we really started out, or I should say, Hawaii was a big part of the beginning of MarineGEO. The Hawaiian Institute of Marine Biology at Moku'uloe, also known as Coconut Island, was the first MarineGEO site in the Pacific and one of the first to join our effort in 2014. And I'd like to tell you a little bit about a major field campaign of research that we did there with our partners in the area back in 2017 to assess the biodiversity of Kāneʻohe Bay. So what we've done also with our Hawaiian colleagues fits into the larger picture of understanding global ocean life, and I'll tell you a little bit about that as well. So the first question is, why Hawaii? The Hawaiian Islands, as you know, are unique on the face of the earth. They are among the most remote islands in the ocean, meaning that there has been a long period of time during which the life forms of Hawaii on land and sea have had a chance to evolve and develop new endemic species and new ways of life. The skilled Polynesian navigators arrived in the Hawaiian Islands more than 1,000 years ago and established the native culture of Hawaii. But the remote islands, especially in the northwest of the current Hawaiian Islands, have been little influenced by people even to this day. And this is a major reason why this region has been protected as a national monument and is the largest marine protected area in the United States. That is the Papahanaumokuakea National Monument, which is over almost 140,000 square miles of the Pacific Ocean. So the first step that we take in trying to understand these ecosystems and how they work and protecting them is knowing who the players are. Who are the marine species that occur in these areas and how do they work together to create the reef, for example. So this was the reason that we and our partners in Hawaii were excited to join forces. And we began that with a biodiversity assessment in 2017 that we called Ola Ike Kai, meaning that life emanates from the sea. It was a great honor to begin that campaign by meeting the Aina-based community of Windward Oahu and the kupuna, the elders of this community, during a welcome ceremony. Kahu Aaron Mahi and other kupuna welcomed our visiting team and blessed and granted the MarineGEO team permission to enter Kāneʻohe Bay, which was a very moving and welcoming beginning to our visit and research. We had a a really good conversation, talking story about our goals, the importance of maintaining the heritage sites, and how together we can better understand Kāneʻohe Bay's marine life and the consequences of ecological change. We also had some great food and had a wonderful time together. So we had about 100 people in our MarineGEO team that came to Kāneʻohe Bay, coming from 24 institutions around the world in 9 countries. And it was especially cool that we had a big group of students from the local area, from the University of Hawaii at Manawa, from the local community colleges, and even high schools. And the students were partners who contributed at every stage of the science, in the field, in the lab, even at night, as you can see here. And so what I'll do now is just briefly show you a bit about what we found and some of the statistics there. So we moved out across the bay and its many diverse habitats and surveyed all kinds of organisms, fishes, invertebrates of all kinds, sponges, corals, seaweeds, and so on. And what these maps show are the diversity of species of several different kinds around the bay. The red spots mean this is places where we had found lots of species, and the green dots were where there were fewer species. So we found over 100 species of fishes, about 1,400 species of marine invertebrates, animals without backbones, including 300 species that had not been recorded from that area before, and more than 60 species of limu, or seaweeds. What's especially interesting to me, I think, as you look at these maps, is that the red spots are in different places for the different kinds of animals. So this shows that having the diversity of landscapes, patch reefs, open reefs, seagrass beds, and so on, within a seascape like this is important in maintaining that high biodiversity. So most studies of coral reef biodiversity focus on corals and fishes for some good reasons. Corals provide important physical habitat for everyone else in the ecosystem. And fishes are important partly because they provide food for us, but also because they're ecological engineers that can prevent algae from overgrowing the reef. And they are responsible for structuring the community of organisms living there. But there is also a whole world that doesn't meet the eye. And if you look at the coral on the left here, when you go down inside that, you find an amazing diversity of small animals. And this is just a sampling of some of those 1,400 species that were found there among the coral and the rocks and burrowing in the sand. The important thing about knowing these species is it can help us inform better care of our living resources. And I'll show you just a few examples. Within that group of species that we found during this assessment, we found several different kinds of species. This thing up on the upper left is a sea cucumber. This was a new species that had never been seen before, or I should say never been formally scientifically described, which is kind of amazing because it's a big organism that's just lying around on the bottom out there. We also found some new invasive species, including this crab in the middle, and also the seaweed that was beginning to overgrow the reef in parts of the bay. And then finally, we found some species that were thought to be extinct in Oahu that had not been seen in this area since the ice, or had only been found in Ice Age rocks. So lots of cool new things coming out of the assessment. So what do we do with that data? Again, a large part of this, in addition to just the discovery of amazing new life, is to inform the larger goal of protecting marine life. And so here, I'd like to tell you a little bit about how we are doing that in MarineGEO across a larger area. So we've learned over time that the best way to ensure the security of biodiversity, the living air and water and ecosystems that we depend on, is to protect them from human impacts. And for example, a major effort has gone on in recent decades to protect fishes and other systems from fishing. We've learned that this is the most effective way to safeguard these habitats and our fellow living creatures. Marine protected areas that are protected from fishing have more fish, bigger fish, more biomass, and more species. So much of the science now is about how to site these properly in ways that protect biodiversity, but also allow people to have their livelihoods. And we've embarked on a new project that is intended to essentially map the marine biodiversity throughout United States waters and understand the degree to which it's protected. So the NOAA Marine Protected Area Center has estimated that 26% of the US exclusive economic zone is protected, which sounds good, but only 3% of that is in fully protected areas that where the animals do not have to suffer from fishing. Interestingly, half of that area is in the remote Pacific Islands, including Hawaii. So how do we use that kind of, gain that kind of protection for other places in the world? This is why we have started the Marine Biodiversity Dialogues project that Smithsonian MarineGEO is co-leading with our colleagues from the University of Queensland in Australia. This is has been sponsored by the Lenfest Ocean Program and the National Marine Sanctuaries Foundation. And the goal of this project is to evaluate what biodiversity is protected in US waters and, well, to evaluate the extent of marine life in US waters and the degree to which it's protected so that we can inform better management going into the future. So we did this by bringing together an expert group of scientists and developing criteria that we can use for this mapping, which is underway now. So I'd just like to close by noting that another really critical way of protecting nature is education and outreach and sharing the love that we all have for marine life and the wonder, and making sure everyone knows how important and beautiful marine life is. And this is where public aquaria like the Waikiki Aquarium and zoos and museums can really help lead the way. So we are very proud to be part of these efforts and working in Hawaii with our many partners, and very happy to speak with you as part of the Waikiki Aquarium's Distinguished Lecture Series. And before I turn it over to my colleague, Mary Hagedorn, who will tell you a really amazing story about how science is being used to help protect reefs, I just want to close with a quote from President John F. Kennedy from many years ago, who said that I really don't know why it is that all of us are so committed to the sea, except I think it's because we all came from the sea. And when we go back to the sea, whether it is to sail or to watch it, we are going back from whence we came. So I thank you for your attention, and we will be happy to answer questions. Aloha and thank you.
I'm Dr. Mary Hagedorn, and I'm a senior research scientist at the Smithsonian Conservation Biology Research Institute and the Hawaii Institute of Marine Biology. And today I'm going to tell you about using advanced reproductive technologies to help save coral reefs. Now that's quite a mouthful, and I'm sure some of you have experienced, or you have friends or relatives who have experienced human in vitro fertilization clinics. And we've more or less taken those same technologies and applied them and adapted them to coral reefs. And today I'm going to tell you about how we've done that, but also how we've applied those technologies to conservation and restoration of coral reefs. But first, I'd very much like to thank the Waikiki Aquarium for this opportunity to talk to you today, and to thank some of my funders and collaborators who've been part of this work for many years. So as I said, I'm at the Hawaii Institute of Marine Biology, and for many of you here in Hawaii, you know Kāneʻohe Bay is this amazing ecosystem that Emmett told us a great deal about in terms of the biodiversity assessment. It's about 45 kilometers of patch reefs, fringing reefs, and barrier reef, and it is an amazing natural laboratory. And it is here that we've created all the technology and applied it to first Hawaiian species, and then have taken it to the outside to other places in the world to quickly apply it. So Hawaii is our laboratory for this conservation work. Coral reefs are among the oldest and most diverse ecosystems on the planet, but in every ocean they are under siege. We live in an ocean planet where our seas produce more than 50% of our oxygen. And within our oceans, coral reefs are one of the most important ecosystems. And that's because coral reefs provide homes for 1/4 of all marine life on the planet. So when you think about it, they're very tiny in terms of the landscape that they take up on our planet, but they are enormous multipliers in terms of how they service our planet and the animals and ecosystems that they provide for. They are also a source of potential medicines such as antibiotics and cancer and HIV AIDS drugs. And if many of you remember some of the tsunamis we've had in Hawaii, you'll know that reefs form natural storm barriers for coastlines, cities, homes, and protect more than 200 million people worldwide. So they're really important in terms of saving our homes and our cities. About 500 million people worldwide depend on them for their livelihood, whether it's tourism or fishing or whatever. Reefs contribute $350 billion annually to the global economy. So they're important drivers of our financial systems. But oceans are, as I said, are under siege, and some of the stressors are both local and global. And I have just 2 images here to show you some of the local stressors. Here you can see sedimentation coming out from a river from space. And this is something we suffer from in Kāneʻohe Bay quite a bit. It's has a lot of sediment from our rivers and from people building along the bay, but also overfishing. Reefs need fish and fish need reefs. And when we overfish, we actually impact our reefs. But a really important stressor is global, and we are overusing fossil fuels and producing greenhouse gases. One of the more important greenhouse gases that we're producing is carbon dioxide. This produces both ocean warming and ocean acidification. And these in turn produce coral stress, coral bleaching, disease, decalcification of the coral skeleton, reproductive losses, and potentially death to the coral. So the state of coral reefs is not that great right now, and as local and global stressors are impacting reefs, as I mentioned. But bleaching is becoming more frequent across the globe. But if you think about what has happened here in Hawaii in the last 10 years, you know that in 2014 and 2015, we had global bleaching events. And in fact, people could see the corals bleached from H3 coming down to Kāneʻo. You could just see the white skeletons. We also had another one, a minor one in 2019. So the bleaching events are increasing in their frequency. As a result, many species of corals are now threatened. 50% of the Great Barrier Reef has died since 1985, and 95% of reef-building coral in the Caribbean have vanished. So this is not a good place to start when thinking about coral reefs. However, the rest of this talk will hopefully be optimistic. So what we think that cryopreservation can help save the genetic diversity and species diversity of coral reefs. And that's really kind of critical when we think of coral reefs, is saving diversity and genetic diversity. So what are some of the solutions and challenges that we're facing for cryopreservation in terms of coral? And first we'll talk about reproduction. And you'll see that I have a panda here. And people love pandas around the world, but they only they only reproduce once a year for about a week. And coral are much the same. They only produce a few nights a year. And so we have very limited time that we can work with a species. So it's about 12 hours that we have a year to work with an individual species. Sometimes they'll spawn 2 months in a row, 3 months in a row, like they do here in Hawaii, but like on the Great Barrier Reef, it's only once a year. They're in very remote areas of the world, so they're hard to get to, and sometimes they can be hurricane prone, and I have been thrown off reefs in Belize because of hurricanes. And they're also stuck to the bottom, they're immobile, and so all of them must be synchronous. If they cannot synchronize the reproduction, then they start losing their reproductive ability. But they're also hermaphrodites, and that means that they produce both eggs and sperm, unlike pandas, which are either a male panda or a female panda. The coral are both males and females. So one of the more important take-homes from this talk is that coral are animals and that they reproduce. So if you can remember that, I'll be very happy. So here you see an individual coral polyp. It looks like kind of a flower with tentacles, and it can divide and produce the whole coral colony. So they're all genetically identical. And then when they produce this thing called an egg sperm bundle often. And this is think of it as a cluster of grapes, which are the eggs, and then wrapped around sperm. And here's what it looks like when the egg sperm bundle breaks apart. You can see the pink eggs and the white sperm. So now I'm going to quickly show you what corals look like when they reproduce. This is a huge egg sperm bundle. They're filled with fat. The eggs are very large and they float to the surface. Sometimes they'll break up ahead of time, but usually they'll go to the surface. And as the eggs break up, the sperm packet breaks up, and the sperm must swim away and fertilize a nearby neighbor. They cannot self-fertilize, or they don't often, I should say, self-fertilize. But there are other modes. This is a mushroom coral that you're going to see here, and they are sequential hermaphrodites. So they turn from males to females every year. And this is a brain coral, okay? So here's our lab team here in Hawaii, and many other people here are responsible for the work that you'll hear and see, in particular John Daly in the bottom right and Jessica Meister in the center. So we can freeze and thaw coral sperm successfully and have trained people all over the world to do this. So it really has been a very international and national collaboration to allow us to freeze coral sperm. And we've developed a lot of the technology to do this, whether it's the actual method, but also the equipment to do it. And here you see some of our earlier efforts on the left, which is flip-flop material, and you see some aluminum cryocanes and the plastic tubes that hold the sperm. And that has now evolved into a 3D printed device that goes into a thermos. And our goal really in doing this was so that it would be inexpensive and easy to do, so that any lab around the world could create their own coral freezing racks and then freeze coral sperm. So around the world today, we have 48 species that have been frozen, and I don't expect you to read the species, but if you just look, you can see that we have 10 species from the Caribbean, 4 from Hawaii, 4 from French Polynesia, 4 from the Gulf of Mexico, and 26 from the Great Barrier Reef. So we've got, of the 1,000 species, we've got about 50 so far. So what can we do with coral sperm? We can freeze it, and it will stay frozen but alive for a day, or a year, or tens of years, and theoretically even hundreds of years. So it can stay frozen as an archive, but it's important also that we use our resources to try and help to restore coral reefs. And here you see an example of a really lovely cryopreserved, a coral that was produced with cryopreserved sperm. It's a Cropperocytheria, and it is, you can see the skeleton and the 3 tentacles from the 3 polyps, and it also has its internal symbionts, which are the things that give coral color. They're algae that live inside them, and those are those brown specks. So this is a completely developmentally normal coral from cryopreserved sperm. Now, in addition, besides just producing more coral to diversify reefs, people want to actually do this thing called assisted gene flow. And it is really just a fancy word for selective breeding. And farmers have been doing this for millennia, where they'll take a really good reproducer or a really good milk producer, and they'll cross them with other individuals to get the best traits that they want. But for coral tartar, as I told you, they're stuck to the bottom. You can't move them like you would a cow or an elephant. And so you have to actually move the sperm. And since they are only produce sperm for a few nights a year, that becomes very tricky. So cryopreservation is critical to this process. I'm going to show you a very short video of what we've done with assisted gene flow. Coral are a fundamental ecosystem in our oceans. We really need to save them. They're really critical for life on Earth. My name is Marine Hagedorn, and I work on coral conservation and preservation. We've been applying human reproductive techniques like freezing sperm to coral, and it allows you to preserve those cells for a year or hundreds of years. We are going to thaw sperm that's been in our banks for over 10 years and use them on fresh curacao eggs to create embryos that wouldn't occur normally in the wild. For this particular species of coral, which is elkhorn coral, there's a western population, which is like Florida. There's a mixed population, and then there's an eastern population, which is here in Curacao.
If the corals do fertilize, then we have a whole new tool for coral restoration, which is the ability to bring different genotypes from one region to another.
And that's what the goal is here, is to help diversify the population and to give us cues as to whether those newly created reproductive organisms are going to be better at adapting for the future.
Around the Caribbean, we've lost something like 98% of this coral species. And in Florida, it's even worse than that because we're finding that there aren't very many genetically distinct individuals. They're one of the most important species in the Caribbean because of the structure and the shape they make. Also because they protect shorelines from waves, they create habitat for fish to hide underneath, and they're also a powerful species because when they do break apart, they can attach back to the reef and make more colonies. This species has bundles of eggs mixed with sperm, and it holds them in its mouth, and then we know in the next 10 or 20 or 30 minutes that they're going to release them all at the same time. We have all sorts of nets and contraptions that we use to collect the eggs once they're released from the colony. Then we have to get back to shore.
That gets transported into the laboratory. We'll clean the eggs very well, and then we'll begin adding sperm.
If it all goes great, the eggs go from 1 to 2 to 4 cells and start dividing and forming the larva.
We'll put them into water bottles, and we'll transport them to Miami, and our partners, Florida Aquarium and Moat Marine Lab, will meet us there. And they'll begin the process of settling those larvae.
This is it. Here's all of our coral babies. They're all doing fantastic. I was super scared when I first got these things that they were going to be so totally different that they wouldn't work out. But thankfully I was wrong. They're extremely healthy. They're growing extremely rapidly.
We couldn't ask for a better result than what we're seeing right here. This is amazing, it really is.
Calling that a win.
Absolutely.
It's a lot of work that went into these little guys.
Yeah. To get to this place today, it's taken us 15 years. It's super important that we make really robust banks to conserve the genetics in as good a state as we can. This not only can work, but it can work in the kind of numbers that can be an assistance for restoration. Okay, so our coral larvae are still alive 2 years later, and they are being tested for heat resiliency. And our hope is that at some point they may go back out onto the coral reef tract to help diversify the coral there that are really genetically depauperate right now. But there's a lot of conversations with managers and local people, whether they're in Florida or Curacao, to really see if that's going to work for them. But in addition to sperm, oftentimes when you go to an IVF clinic, you may want to have your eggs frozen, or you may want to have your embryos frozen. And for coral, that was a really huge challenge. And here on the screen, you can see these beautiful images that Andrew Haywood took of coral larvae and dividing in 2 and 4. and they're extremely large and filled with fat. And so the challenges for coral, you can see a coral larvae here, and they are about 7 or 8 times the size of a human embryo. And so they're much bigger just in terms of their diameter, but their volume is massively bigger. And so they are also very sensitive to chilling. So when you bring them down to like 0 degrees, you can see that they get damaged. And the image on the right in that yellow panel shows you what happens when a larvae is just exposed to 0 degree temperature, no chemicals, no anything, just seawater, for about 30 seconds. They fall apart. And the other thing is that ice formed upon warming because they're very large and they have a lot of water in them. So these were huge challenges that we did not, we really took a lot of pains to try and overcome. It took us many, many years. And when John Daly came as a postdoctoral fellow, he started using lasers to warm the larvae. And what this did is it warmed the larvae at millions of degrees per minute, actually 7 million, and it stopped the formation of the ice crystals. And it was the ice crystals forming during warming that was really the damaging part. And so I'm going to show you a little video of what he does. We take a pipette with some larvae, and we put it on a drop on a blade, and there's gold in there. There's gold nanoparticles, and those are the radiators of the laser warming. They re-radiate and warm the larvae. So now we put it in the holder, and then the 10 or so larvae go into the liquid nitrogen. They're in a drop of cryoprotectant solution, which is like an antifreeze, and they freeze solid. There's no ice at this point. And so when it flips up, you'll see a red beam, which is the laser, and it warms it at millions of degrees per minute. So the way John knows that the larvae are doing well is he tests them for swimming. And here you can see some coral larvae that are swimming, and he gets about 50% of his larvae that survive post-thaw. Now, the other thing you want to check besides whether they'll swim is whether they'll develop normally. And two really important criteria for that are whether they'll settle and whether they'll take up their symbionts. Now, the symbionts are the brown dots that you see here with this at the end of the yellow arrow. And if they cannot take up their symbionts, they will never be developmentally normal. We call them non-competent. But this laser worm larvae is, and it has taken up its symbiont. So we know that when we laser worm these larvae, they will grow to be normally developing larvae that can settle. But when John does this, he may in a day get about 300 larvae that he can do with this laser warming. But we need to actually go and try and do much more. If we're going to use this as a restoration technique by, putting larvae back out on the reef, we need to produce thousands of larvae at a time. And that's because coral die for a business, really. A coral may produce 1 million or more eggs from bundles or larvae, but really only 1 or 2 will settle and grow into an adult. So you have to have a lot of larvae if you're going to produce a small number of adult coral. And so to do that, we mechanize this whole process. And here's a video of that. And these little drops are holding larvae. And we can print them, and they go into the liquid nitrogen, which is right there, and they freeze. And so instead of doing, it on the blade and the laser, we now have this mechanized process. And this is what the Smithsonian has 3 patents, and this is one of them. We hold 2 of the patents for the Smithsonian. And this is called the laser cryo wheel that we developed just with 3D printing in our lab. So here's what they look like, the beads look like. They're a little bit frosty because we had to take them out to take a picture of them on the microscope, but the arrows point out the larvae, the frozen larvae in the beads, and the beads is almost filled with screen. And ordinarily they'd be clear, they wouldn't have sort of ice crystals in them. But you can see that we can produce lots of bees. We can produce about 10,000 larvae an hour now with our cryo wheel. And this is an amazing sort of high throughput restoration technique that we can now apply to coral reefs. So unfortunately, as I mentioned, as global climate change and bleaching is increasing, reproduction is often faltering. And when we have a bleaching event, there's another solution that we are trying to bring forward, and that is freezing coral fragments. And here you see a tiny coral fragment. You can see it's about 1 centimeter in diameter. And the brown sort of rosette looking things are the coral polyps. There's probably about 25 of them on this tiny little fragment. And we're applying some of the most sophisticated engineering technology to be able to freeze coral micro fragments or fragments. And the nice thing about these is we can bank a lot of them quickly, and we can do it around the year. We don't have to wait for, you know, one night a year. We can do it 300 days a year if we want to. And the other nice thing is we can grow these up. We know how to grow these into adult coral. And then those adult coral can go into captive breeding scenarios, which Jamie Craigs has developed at the Horniman Museum recently. So we now have the ability to grow them and breed them in captivity. So this could be an enormous way to diversify reefs by freezing coral fragments. Finally, my last thing that I'd like to talk about today is the World Coral Fragment Biorepository. And this is a new initiative for us, but it's a consortium of foundations and groups working together around the world. And the Florida Coral Rescue has already started this process. And there in Florida, the stony coral tissue loss disease was killing 1/3 of all the species in the Caribbean. And there's only 60 species in the Caribbean, so it was affecting about 22 species in Florida. And so they went out and they collected all 22 species, and they have about 2,000 individuals now that are growing in 18 aquaria around the United States. And their goal is to grow these aquaria to adults, grow the fragments to adults. Hopefully the aquaria have all adults in them. And then what they'll do is they will outplant either the corals themselves, or if they spawn or fragments, and put them back out on the reef. Now, another group is the Great Barrier Reef Legacy Foundation. They are going out in the next week and collecting 200 species from the Great Barrier Reef. And then our third group is the Coral Conservatoire in Monaco. And they are going to be collecting coral in Indonesia. And our hope is that maybe we could work with University of Hawaii, perhaps Waikiki Aquarium, and do a collaboration with the Smithsonian to try and do something with Hawaiian coral. So with that, I would like to point out that biodiversity is the planet's most valuable resource, and it is on loan to us from our children. So thank you very much, and Emmett and I will entertain any questions that you might have.
Thanks, Emmett and Mary. Super. This is the second time I'm watching it, and I learned so much even with this. If you do have questions, we ask that you put it in the chat. If you go down to the bottom of your screen, there's a chat feature, and you can type in your question, and I'll read it to Mary and Emmett, just so that we don't have too much feedback or interference while we're doing the live Q&A. So Emmett, I have some questions, and Mary, I have some questions here already. Emmett, is there a formula that designates like a healthy biodiversity? Like, should there be so many species of fish or invertebrates to designate what would be a healthy ecosystem? And then how does Hawaii compare to the rest of the world?
Yeah, that's a very interesting question. It would be good if it were that easy. But it's not, because the biodiversity varies around the world, and the fact that it's complicated is in some way nice, because that gives me a job to do. But to answer the question, one of the things that we're learning about and discovering is essentially how much diversity is needed for particular kinds of ecosystems. So, an estuary in the Arctic may be doing very well with a very small number of species, whereas a coral reef in the West Pacific, for example, might have hundreds of times that number of species. And that's because species are doing different jobs in those areas. However, we now have enough data from around the world that we're beginning to understand, what numbers of species and more importantly, what kinds of species are necessary for different ecosystems to function properly, to be able to resist disturbances and continue to provide for people. So there's not really a single formula, but there are ways of understanding what is needed in particular systems. And you asked about Hawaii also. You know, Hawaii is a very unique place, as I think Mary and I have both made the point, being in sort of isolated from other sources of marine life in the Central Pacific. It's evolved to work. First of all, it's evolved quite a few species of its own that only occur in Hawaii, but also has evolved to operate with those specific species. And it does not have as many species as, for example, Indonesia or the Coral Triangle, but they are adapted to working well together.
Thank you. And you mentioned about finding new and extinct species in Kāneʻohe Bay. Have you made similar findings in other areas that you've monitored?
Yes, we have. You know, I mentioned that a lot of those beautiful small creatures are very cryptic, as we say in biology geek talk, meaning that they are hidden down inside the reef, and so most people never see them. But when we came to Hawaii a few years ago, we had specialists from all over the world that knew exactly where to look for the particular creatures that are their specialty. And so we've done that in a number of other places as well. Some of our colleagues have been doing this kind of scavenger hunt for biodiversity all around the world. And pretty much everywhere we go, we find lots of new species. And one of the things that we're doing with those, in addition to collecting them and preserving them so that you know, they're available in museums for others to study, is to take their DNA barcodes, as they say, the unique DNA signature that they have, and high resolution images, and putting those into public databases so that we're able to, so that anyone really will be able to learn from that and understand where these organisms are, how they work in food chains, and so on.
Thank you. Next question is for Mary. Have you identified species genetically of coral that are tolerant to bleaching?
Great question. We haven't yet. Certainly, it would be a great find. There are, I do take that back a little bit, but there's a couple here in Kāneʻohe Bay, that couple species that are just bulletproof, but they're not very common. You know, so they're not the reef building corals, they're very small corals. And so I'm sure there's others around the world. But the thing that's important to understand is that my colleagues at the Hawaii Institute of Marine Biology 50 years ago did some bleaching experiments. This is the work of Paul Jokiel. And he studied, I'm going to say 6 species, and he tested them for temperature tolerance back in the 80s. And you know, looked to see how many corals stressed and bleached and died as a, you know, in response to the higher temperatures. And then they repeated this experiment again just a couple of years ago. And in the 80s, 80%, I'm just going to ballpark it, 80% of the corals died or were stressed in the experiments when they elevated the temperatures by 2 degrees centigrade. And when they did it a couple of years ago, only 20%. So it's very clear that even in Kāneʻohe, you know, corals around the world are adapting. And it's probably something that they can do naturally. When you think about salmon, for instance, salmon live in very cold streams, say in the Arctic, but they also have, the streams can get very warm in the summer. So you have to have enzymes that can work in both slightly warmer temperatures and colder temperatures. And it's likely that coral have some flexibility in that, you know, ability to maintain their sort of tolerance to bleaching. And so, but the other great thing about the bleaching in Kāneʻohe Bay, was that 85% of the corals in Kāneʻohe Bay bleached. And it was shocking. Actually, the bay was completely white. But only 1% of the coral died, which is amazing. And the following year in 2015, the bleaching was as intense in terms of the temperature, but a much lower percentage of the corals bleached and all of them recovered. So even in that sort of very intense example of stress, the corals did pretty well. And I think it's because there had been some smaller bleaching events that weren't really all that big that sort of got them a little bit more acclimatized. So I think that the real risk that we have with bleaching is not that we would necessarily lose coral, although we do. It's that they won't reproduce. When corals stress, they don't reproduce, and sometimes they won't reproduce for years. And so if they cannot reproduce, then they cannot get any more adaptations to how the oceans are changing. So reproduction is really important, and bleaching really impacts that.
And then is there a reason why you don't just freeze the egg and sperm separately? And there are more success that you're seeing in actually freezing coral fragments rather than just the larvae?
We do freeze the larvae, and the larvae actually, now that we know how to do it, is pretty easy, but it took us 12 years to figure it out. Remember, we have 12 hours, a summer to do this. So we have a very tiny window to sort of get this technology to work. But the eggs, so let me give you an example from humans. When human sperm was first frozen, that was in 1960. Human embryos were not frozen well until 1980, and human eggs were not frozen well until 2000. And the reason is because eggs are filled with fat. They tend to fall apart during cryopreservation, and they only have one cell, they only have one membrane that goes around it. So if you nick it or do anything in the process, it just falls apart. And because we have such limited time, sometimes just 2 or 4 hours a night to work on, eggs or embryos, we just haven't taken the time really to work on coral eggs. The sperm and the embryos were just easier.
Can everyone hear me now? Yes? Okay, great. So, sorry about that. So for both of you, can you tell us why you got interested in marine science?
Emmett, you go first.
Yeah, sure. I love talking about that. I fell in love, basically. In other words, when I was like many kids, I was really interested in animals. I didn't grow up near the ocean, but I, was walking in the mountains and, learned about animals. But when we first started going to the ocean, I completely fell in love with, the marine life that I found. I grew up near Washington, D.C., and my aunt, my dad's sister, lived in Los Angeles, all the way across the country. And we would drive across the country having these great adventures. And, the first places where I really saw ocean life was on the coast of California. And I can just remember being mesmerized because the animals were so different than anything we've ever seen. Mary's corals, they're like nothing on land. And that's true of so many of the creatures there. So I fell in love with the marine life. And, when I went to college, I was pretty amazed to find out that I could actually make a living doing this, studying these creatures. And so that's how I got into it. And then, of course, what has happened, I think many of us have a story similar to that, that we became marine biologists because, you know, it's just the life of the ocean is so fascinating. But then, you know, we discovered that all of these challenges that the world is facing, and this has really come up, you know, a lot in my lifetime. And so all of us now, I think, have really had to turn our attention to, you know, how can we understand ocean life better so that we can take concrete steps to protect it and to sustain it into the future. And the work that Mary talked about is a beautiful example of that.
Thank you, Emmett. I think for me, it was very similar, but I grew up on the ocean. So I grew up in Connecticut on Long Island Sound, right across from New York City. But I just had the pleasure of, you know, just being a kid on the ocean. And I just found it infinitely fascinating. I would collect crabs every single day and let them go and collect them the next day. It was just something that I never stopped being fascinated with. And you know, when I went, it's funny, in high school, I didn't take any biology at all. I figured I'd take it in college. And I just always wanted to be a marine biologist. I do remember it when I was 12, I wrote to Woods Hole and asked them if I could come to school there. And they were so polite, they wrote back to me. I remember getting a letter from Woods Hole saying, well, you just have to get a little older and then you can come there. So it's, you know, I think like Emmett, we share this intense love of the ocean, as many Hawaiians do. Everyone in Hawaii loves the ocean. Even if they're not swimmers, they just love being by it. And so I think this sort of passion that starts when you're kids, you know, if your parents are able to help you foster that love, it just, you become a marine biologist.
So Mary, this question is to you. Is there any coral species that, or is there a reason why you pick particular species to do your research on? And is there a way for you to speed up their growth and reproduction so it takes up less time to rebuild a coral reef?
So tell me the first question again. My internet was a little bit spotty. The
Sorry,
first part of the question.
So the first part was, is there a way that you pick coral species to do this type of research on? Are there ones that are more, it's easier to collect samples or what have you?
Yeah. So both are really good questions. And when I first went to the Great Barrier Reef to start a bank there, we tried to get the scientists there to help us prioritize the species. What did they want? You know, how much did they want? We could not get any answers. And I think it's still a difficult question to get answered. So we really work with whatever comes in, you know, and if we can get a species, we'll work with it and we will bank it. I mean, ideally what we would want to do is to freeze different sort of morphotypes, like, you know, boulder kind of corals and branching corals, and, you know, try and make sure that functionally we represent, you know, sort of the ecosystem. In Finding Nemo, that's how they learn to draw coral reefs, by doing long skinny ones and round ones and branching ones. And so I think, I mean, that's not a very satisfying answer, but you know, when you only have like a week to do your work, you can't, and other people are going to collect for you. You can't be ever so fussy about the species that they bring in. So in Hawaii here, we work with, there's only 4 species in Hawaii that we know the reproduction on. We have probably 15 in the main Hawaiian Islands, but the reproductive period of those corals have not been identified. So that's the first part. But to grow them faster, yes, people are doing that now. And the microfragmentation is one of the ways that people are growing reefs faster. And when you cut a coral into a tiny little piece, it turns it into stem cells. And those stem cells just, they grow so fast. And so you can grow, for instance, Chris Page works in my laboratory, and he worked at Marine Lab, as you saw him on the film, but he and his colleagues did microfragmentation of a lot of species in Florida. And then they pieced them together. So they were boulder corals, and they pieced in, in the individual sort of microfrags together, put them on a cement rock. They sheeted that rock within 2 years, and within years they were spawning. So there are ways of increasing the growth of coral in in the ocean, but also captivity is doing amazing things. And this is something that, sort of professional aquarists should be enormously proud of. We can now spawn coral in captivity, and the pillar coral, which is one of the few corals on the endangered species list in the United States, Florida Aquarium brought those into captivity over the last 3 years. And when they first came in, they did not reproduce well. Their reproductive characteristics were terrible. But now in this 3rd year, they're spawning beautifully. Their sperm motility and all the reproductive characteristics have improved immensely. So I really think the role of aquaria in the next 20 years is going to be critical in helping us to restore and to save coral species.
Thanks, Mary. We have time for one more question. And this one is I think more directed towards Emmett. So we have a lot of students attending tonight's lecture. And so they want to know what can they do to help protect the ocean? Like, how often do you do these monitoring activities, and can students get involved?
Mm-hmm. Yes, students can get involved. So in any given place, the monitoring or the research, observation, measurement that we do is usually on an annual basis for the overall surveys. The kind of major effort that we did a few years ago that I discussed, which Mary had a major role in organizing, that was a huge effort, and that's something that we generally do one time at a given site, such as Kāneʻohe Bay. But on the other hand, that brings together people and starts up new projects, and so there's opportunities. What we are working to do as we build up our network is to develop more opportunities for students. So for example, the Smithsonian Institution has internship programs and some support for graduate students, less so for younger students, but we'd like to develop that at our partner sites as well. I think I mentioned that we did have quite a few students working with us when we did the assessment here before. So I guess one thing I would say, I saw a question in the chat box too about going to college and what can students do. And, I would say, to me, it's less important that you find that particular college that everybody wants you to apply for. The main thing is to get to a good college. It can be a community college, a university, but one that's comfortable for you, that seems right to you. And this is really the most important thing, I think, where you can get outside and actually interact with living marine life. To me, that's really important, because that's where the insights come. That's where you learn. The famous biologist from 150 years ago or so, Alexander Agassiz, who was sort of one of the parents of American biology, he liked to say, study nature, not books. And I'm not advocating that you don't read books, but what his point is that if you want to learn about biology, it's going to come from working with the living organisms. So I think there's a lot that's the foundation, and there's a lot that you can do. You get a good school, a good college, anywhere there. In terms of helping, I think the single most important thing you can do when you're eligible is to vote. And in fact, we need scientific knowledge. We've shown some examples of that today, but I think even more important in terms of conservation is understanding people and understanding how to work with people together. And so social science, sociology, psychology, those are really important areas that can be applied in conservation. And I think really law and politics are hugely important areas for conservation. I'm not sure I would advocate anyone getting into politics at this time in history, but I think you understand what I mean. So finding ways to bring the science together, and of course, everyone, no matter what stage they're in, can help by understanding what contributes to sustainability and what is safe to do, and what is helpful for the ecosystems as well as people.
Thank you so much, Emmett. I know we might not have gotten to all of the questions that were posted. So if you want to message me your email address, I can forward your question to Emmett and Mary or put you in contact with them. Or if you're having trouble with the chat feature, you can also use our contact page, and those emails come to me, and so I can direct those as appropriate to Emmett or Mary. So thank you so much, Drs. Duffy and Hagedorn, for presenting your intriguing work to us. It's great to see how Hawaii fits into global ecosystem research and the preservation of coral reefs. Thank you, Emmett. I know it's close to your bedtime, or if not past your bedtime in Virginia. So thank you so much for being with us. And thank you, Mary, also for taking time out of your day to be with us as well.
It is a real pleasure, and thank you, everyone. Getting
Yes.
dark. I forgot to put lights.
Aloha to all.
Aloha,
Yes.
everyone. Bye.
We want to thank our sponsors and partners, Hawaii Public Radio and St. Andrews Schools, particularly Alden and Pang, who helped us set this up, and also to Amelie Katz for doing that great intro. Thank you. If you didn't get to see the lecture from the beginning, we will be posting it on our website, so you can check that out in a few days. Earlier this year, the Friends of the Waikiki Aquarium lost one of our longtime board members, Tim Gard. Tim made so many contributions to FOWA and the community. In fact, this distinguished lecture series was his idea, as he wanted to bring more opportunities for education and outreach to more people out there on marine ecosystems and issues. And so it is our honor to rename this program the Tim Gard Distinguished Lecture Series to keep his legacy going. Please check out our website at fowaquarium.org for past lectures and other resources and our future activities. You can also contact us with questions or to make a donation on our website as well. So mahalo nui loa, and thank you for joining us.